Almost 100 years later, X-ray crystallography is still used for the same purpose. The molecules are more complex and the equipment more sophisticated, but the basic principle remains the same. The molecule being studied is converted into a crystal, which locks its atoms into a uniform three-dimensional structure. X-rays are then beamed at the crystal. As they pass through, they are bent or diffracted by the clouds of electrons that surround these atoms. These scattered X-rays are recorded on a film or photosensitive receptor as a pattern of dots that is unique to that particular molecule.
And we can figure out from the direction of the scatter where the electrons were that produced that scattering pattern, and from that, the shape of the molecule.
By the early 1950s, X-ray diffraction of DNA revealed some tantalizing clues about its structure. But the X-ray dots were like tiny peepholes through which only certain features of the DNA molecule were visible. One feature the dots revealed is that the nucleotides in a DNA chain join together to form a backbone of alternating sugars and phosphates. The dots also showed that the DNA backbone curls around its axis to form a corkscrew or helix shape. The dots also suggested that a DNA molecule is composed of two such helical backbones winding around each other. The resulting structure is called a double helix. However, the X-ray photos were silent on the third component of the nucleotide, the 4 different bases. Because of their variability, they scattered X-rays too randomly to form dots. They therefore remained invisible. But the bases had to be within the double helix, attached at right angles to the sugars. Opposing bases must somehow bind to each other, gluing the 2 backbones together. It had long been known that the 4 bases come in 2 different shapes. A and G are larger 2-ring structures called purines, and C and T are smaller 1-ring structures called pyrimidines. Scientists also knew that identical bases could join together side by side through hydrogen bonding. But such base pairs are either too big or too small to match what they knew was the diameter of the double helix. Also, such base pairs could not have produced the very regular shape suggested by the X-ray photos, which resembled a smooth-walled cylinder. To fit them in, the cylinder would have to bulge out for the larger purines and shrink in for the smaller pyrimidines.